activity to behavioral phenotypes across subjects, continuing the theme of individual differences here. So we know, obviously, that movies evoke similar brain responses across people, but we also know, especially from what we've just heard, that they also evoke some really interesting individual differences. And movies are also a lot of fun to work with because they lend themselves to a variety of different types of analyses. So far we've heard a lot about functional connectivity, and that is one really interesting thing you can do with movie data. But there's a lot of other stuff, too, that you can do with movies that you can't do with rest. And so I like to think of these as sort of a spectrum from treating the movie as a more time-locked or task-like paradigm all the way down to a less time-locked or more rest-like paradigm. And so one thing that we could do towards the more task-like end of the spectrum would be to model different features of the task and just use these as regressors, similar to what we would do in a traditional fMRI experiment. We can also do things like intersubject correlation, and then of course down here on the other end we have things like functional connectivity. So the main goal of the project that I'll be telling you about today is we wanted to know which of these three approaches was most sensitive or best pulled out variants related to a behavioral phenotype of interest. And so the data that I'll show you come from actually the other movie that was included in the OHBM data set, which is The Present. So this is a very cute animated short about a boy and a dog. We're working with 68 subjects here, mostly chosen for data quality. And the phenotype that I'll be focusing on here is the SRS, or the Social Responsiveness Scale. As others have mentioned, this is a movie with social and emotional themes, so we hypothesized that it might pull out variants related to social function. And so you can see some example items on this scale here, and the important thing to remember is that higher scores here indicate a greater impairment. So here was the distribution of SRS scores in this sample. Miraculously, SRS was unrelated to either age or head motion in this particular group. And for the analyses I'll show you today, we just did a median split, so we had a low SRS and a high SRS group. Now I'm going to talk a little bit more about those three methods I mentioned earlier and some of their advantages and disadvantages. So the first thing we were interested in was using these feature-based regressors, so extracting features from the movie itself. And so this will be very familiar, just akin to a regular traditional task-like analysis where you're using events or a continuous regressor and you're looking for voxels that show increases in activity. The advantage of this approach is that it's time-locked and it's directly interpretable. So you know what you're modeling and you know exactly what these voxels are responding to. A disadvantage here is that you have to make a lot of assumptions about the structure of the task and also the hemodynamic response. Another thing you can do is intersubject correlations. So the idea here is that if you look at the same voxel in two different subjects' brains, and that voxel is doing something similar over the course of the movie, you can infer that it's somehow involved in responding or processing the stimulus. And this is a nice approach because it's still taking advantage of that time-locked nature of the task, but you're making fewer assumptions. And one of the challenges with ISC is that it's an inherently pairwise metric, so it's harder to model at the single subject level. And finally, we have functional connectivity. There's obviously many flavors of this type of analysis, but the general idea is that instead of looking at magnitude of activity in one region, you're looking at how different regions fluctuate together. So the advantage is that you're capturing interactions between brain regions, but the disadvantage with movie watching data is that it's not time-locked and it's somewhat harder to interpret. So just a little more detail about how we actually operationalized each of these three analyses using the OHBM data. So we've pulled out 6 features from the task that I've arranged in sort of a loose order from lower level to higher level. So we modeled just auditory information, so volume. We modeled low-level visual changes in luminance, also motion via optical flow. I mentioned that the movie has both a boy and a dog, so we had 2 regressors for times that the boy was on screen and times that the dog was on the screen. And our highest level regressor was emotional faces, so anytime there's an emotional face, either positive or negative. For intersubject correlation, you can think about this as at every single voxel, we can get a matrix, which is subjects by subjects, telling us how strongly correlated that voxel is over the course of the movie in different pairs of subjects. And so for purposes of this, of course, we're interested in these phenotypic effects, so we can start to look for voxels that are more synchronized in pairs of low or pairs of high SRS participants. And finally, for functional connectivity, again, there's many ways of doing this. Here, to be most comparable to the other 2 approaches, we just did a voxel-wise intrinsic connectivity approach where we're just looking at the mean correlation of each voxel to all of the other gray matter voxels in the brain. So first I'm going to show just the main effects of each of these. So there's no effect of SRS here. We just sort of wanted to get a handle on and see that each of these three analyses were behaving in the way we would expect. So the results from our 6 regressors, we saw, happily, we saw auditory cortex lighting up to audio information. We saw visual occipital regions for the luminance regressor. Same thing for motion, also with some interesting deactivations. For both the boy and the dog regressor, we see visual occipital extending into the more temporal regions, and same thing for emotional faces. These generally look somewhat reasonable. For functional connectivity, again, this is all at the group level, so we're seeing that there's a lot of connectivity throughout the brain, but the visual regions tend to be more strongly connected, which again makes sense given that this is a visual task. And for intersubject correlation, similar to what others have observed with these types of naturalistic stimuli, we see that the movie evokes a lot of correlation across all of the subjects, not only in primary sensory cortices, but also extending into higher order association regions of the brain. But of course, what we want to know is not so much what's consistent at the group level, but what varies with our behavioral phenotype of interest, which is SRS. So here I'll show you the results from the high versus low SRS contrast. And these are the results from the regressors. And I'll just mention these are a fairly liberal threshold. They are cluster corrected, but we just sort of wanted to see what was coming up here and which of these was going to give us the most sensitivity. And actually, the regressors did not show huge differences between the high and the low groups. So the maps that are grayed out here didn't actually show any significant differences between high and low SRS. There are some interesting things going on here, although generally small. One of the more interesting findings from this, it's very hard to see, but actually the fusiform gyrus is more strongly activated to emotional faces in the lower SRS subjects, which is the direction that we would expect because these are the children that are better on social functioning. But overall, I would say that these results are fairly unremarkable in terms of pulling out differences with SRS. Functional connectivity actually didn't show any, at least in the way that we've analyzed it here, didn't show any group differences. But interestingly, ISC actually pulled out a number of regions that were more synchronized in the low SRS group versus the high SRS group. So all of the blue regions in this map here had a more stereotyped response across pairs of low SRS participants versus pairs of high. And again, that's the direction we would expect. So it seems like the lower SRS children are all sort of synchronizing with one another, while the children that are worse on social function have more idiosyncratic responses that are less similar to each other as well as to the low SRS group. So it seems, at least from these preliminary analyses, that ISC is the most sensitive at a whole brain level to these types of phenotypic differences that we're interested in. So to provide you with some interim conclusions, we're still actively working on this data set, but we have seen so far that ISC seems to be more sensitive than either the feature-based approach or functional connectivity to these phenotypic differences in social function. And this is really interesting because it suggests that there is some, what I'm calling zeitgeist, for lack of a better word, of the stimulus that we're not capturing with these particular 6 regressors that we've chosen to look at so far. And maybe there's others that we would see these responses with, but at least the encoding models that we've tried don't seem to be capturing these differences as well as just looking at other subjects' brain activity. So that's a really interesting point, I think, and that brings up our next steps, which are we'd like to try to find out what's actually driving activity in the regions that were identified in the ISC analysis. And the ultimate goal of this line of work would be to predict behavior in novel subjects based on intersubject correlation. So with that, I'd like to thank everyone, especially thanks to the Child Mind Institute for collecting this awesome data, and I'd like to introduce